For all of the people out there who have had children, here's just a bizarre question to even think about. If you could have, would you have hit pause on the embryo's development? So the fertilization of the egg has happened. It's going to develop into a fetus and ultimately into a baby. But imagine you could pause it and be like, no, not yet. Let's wait a little bit.
Why would I do that?
I don't know. Maybe the timing's off or something.
We can do that.
That's the crazy.
Animals do it.
Yeah, animals do it, but humans apparently can or could. That's the crazy implication of some Max Planck Institute research. It ultimately has some potential applications for in vitro fertilization. We don't want to get anyone's hopes up or to give false hopes that will happen soon, but it's worth finding out how to do this in human beings and why it might ultimately matter.
But first of all, in a petri dish.
In a petri dish. And for the rest of this interview, we're going to use the word embryo as if we're talking about actual human embryos. Just a big caveat here. These are not real human embryos. These are generated from stem cells, from pluripotent cells, and for all intents and purposes, act like a human embryo.
Science Unscripted.
So my name is Aydan Bulut-Karslioglu. I'm a researcher at the Max Planck Institute for Molecular Genetics in Berlin, Germany. And together with my team and our collaborators, we recently found out that human embryos have this inherent capacity to self-regulate their developmental timing.
Aydan, you just said modulate with regard to an embryo's development. The way I understood it was that you could hit pause on embryonic development. Did I understand that right?
Exactly. And we can do that by altering the activity of a protein called mTor, which regulates cellular growth. And by reducing its activity, we showed that we can slow down the development of human embryo models.
Let's get to humans in a little bit. First, let's stick with animals. We know that 100 or so animals do this naturally.
Wait, what's one of the animals?
Yeah,
let's go
animal. Let's go with one example and take us through what happens.
Exactly. I will give you two, if you allow me. One is, for example, roe deer, which is very common in forests of Europe.
Roe deer?
Like a white-tailed deer?
Roe deer.
Oh, yeah, a deer, yeah.
Exactly. And these animals mate in, actually, I should get this fact right, I think in the fall. And after mating, the sperm get fertilized to initiate the developmental programs of the species. But normally, if it goes on like this, the pups or the fawns are going to be born over the winter, which is less than ideal for further prosperity of the species. So what happens is that once the embryo passes through these initial few days of development and reaches the stage called blastocyst, then development is slowed down. So the embryo hits the pause button and takes a break from development, and it's reactivated actually 4 months later in this embryo to continue and to eventually give rise to the live progeny in the spring, which offers better conditions.
Okay, so the fertilized embryo is inside the womb of the female deer, the doe,
Exactly.
and senses chemically somehow. There's a chemical signal in there. Oops, it's too cold out there. I should slow down.
Well, sort of. This signal is reaching the embryo via the maternal system, because this is all hormonally regulated from the maternal side. And the maternal side tells its own system, essentially the uterus, that the development should not proceed at this stage. So the uterus now does not become receptive, the embryo cannot implant, and just resides in utero, in the uterine fluid, without implanting for a few months until it can do so at a later time point.
You wanted to mention a second animal? What was that?
Exactly. I wanted to mention a second animal, which could be the mouse in this case, because this is completely differently regulated. In the roe deer, I mentioned that this is seasonal, and it always actually works the same way, every mating season for these animals. In the mouse, it's more acute. meaning depends on whether there's scarcity in the environment. So it's on-demand pause button, if you will. And what that means for the case of the mouse is that, of course, food is not a given thing. And if the mom is starving but is pregnant at the same time, then that risks the progeny. And at that point, then that mom can tell chemically by signaling to the embryos that this is not an ideal time to develop, and the development of that litter, again, can hit a pause button for a few days to a few weeks until nutritional sources are better.
And in both these animals, this process happens naturally. In human beings, it does not happen naturally, but you have somehow managed to make it happen? Fill us in?
Exactly. I have to say that it's very important to say we did not actually directly make this happen on human embryos. We used models of human embryos which very closely represent the human embryos, but these are not embryos. I think this is very important to say at this point. Yes, otherwise you're correct to say that we could make this happen on human embryo models and stem cells. And the reason is very simple, actually. We know that a lot of mammalian species possess this capacity. And we have worked for years now in our lab with a system where we are able to put mouse embryos into pods like this in the incubator in the lab. And so we said a lot of regulatory events are actually conserved across mammals, meaning like they are applicable to a lot of different species. So the idea was very simple, and we essentially asked whether the same protein, the mTor protein, can also regulate the timing of human embryo models, just like it does in the mouse.
And when you say that you're modifying the mTor protein, is that in the uterus or is that in the embryo? What are you changing to make this happen?
So this is now entirely outside the maternal system, because anyway, in the human, we cannot do that. We don't want to do that. But also on mouse embryos, we collect the normally developing embryos from the uterus. We put them in cell culture in the incubator in the lab and culture them with a so-called mTor inhibitor to reduce the activity of this protein. And that puts them into a paused state. And now with these blastoids, which are human embryo models, we did the same. We took these and cultured them in cell culture in the incubator together with this mTor inhibitor and showed that we can actually alter their developmental timing.
The reason why animals do this is you said self-preservation, or to make sure that the progeny survives. What would be the application of pressing a pause button on embryo development for human beings?
So here we should focus on technological applications, so not in vivo or not within the uterus, of course, but IVF has offered huge chances of success in human reproduction. It has helped a lot of families, and it's a very successful operation, has been recently developed within the last decades, actually. But there are also cases, even within this success story, where we don't understand why reproduction fails. And we guess that some of it comes from a mismatch of the timing of embryos' development and the maternal system. So here is where we hope we can make a difference in terms of gaining the extra time to perhaps time things better from both sides. Or perhaps even to characterize the embryos better, because once you start embryonic development, starting with an egg and a sperm, you only have a very limited time frame to complete these first stages and to transfer the embryos, if they're not going to be frozen, into the mom. Now we hope, of course, with some further work and development, that we can gain more time within that time frame.
So I'm going to wildly speculate here based on what you just said. 10 years from now, 15 years from now, a couple walks into a clinic, they want to get pregnant, they're having issues, they're struggling, it's not happening. And to possibly increase their chances of getting pregnant together, they might take an mTor protein therapy that would adjust the amount of time where that embryo would be viable to attach. That might be like a step before IVF. Is that completely off?
Time-wise, it's hard to speculate when that could happen. And this, again, was the very first step of even showing that that is possible. So I think we also have to be careful about developing it enough before offering therapies. But yes, essentially, if it gets to that stage, the offered therapy could be potentially to alter the timing of the embryo in the clinic before it is transferred to the mom, so that one just has more time for characterization and for better matching of the embryonic and maternal systems.
And that was Aydan Bulut-Karslioglu talking to us from Berlin in that case. And if you're interested in reading up on the public or looking at the publication, this is mTor Activity Paces Human Blastocyst Stage Developmental Progression in the journal Cell.
And she wanted us to reiterate at the end that we're not working with human embryos. It was in a petri dish. These were what's called blastoids, not at all actual human embryos. And she also wanted us to mention, do not get your hopes up at this point. If you're hoping from hearing this that it's going to help you with IVF, the research is not at that point yet, but it could be one day, essentially.
Yeah. This is, I think, from her perspective, basic research. It's interesting. It's important. It leads to lots of interesting speculation, but it's just that at this point. Anything else to say? Tell us. Say it to us. We're SU@DW.com.